Hello and welcome to this video covering the most common styles of exam questions within the transport across membranes topic. We will look at the structure of cell membranes, different types of transport and the two required practicals that link with this topic. To get the most out of this video, I would suggest completing the exam style question worksheet in advance. Our first question relates to required practical 3, which is the water potential required practical. And in this experiment, the student has taken cubes of carrot and submerged them into different concentrations of sodium chloride solution. They weighed their cubes of carrot individually, left them in the solutions for 24 hours, and then reweighed their carrot cubes after they'd taken them out of the different solutions. And the first question asks you about temperature and how you would ensure the temperature was constant at 25 degrees C throughout the experiment. And the key thing here is that you look at the number of marks available. So there are 3 marks available. So it's really important for this one that you fully explain your answer. So talking about using a water bath only gets you 1 mark. To get more marks, you need to say how exactly you're going to keep that temperature constant. And you need to be checking the temperature. So at regular intervals, you need to use either a thermometer or a digital sensor to double check that your water bath is actually at 25 degrees C. And then if it moves away from that given temperature, how are you going to correct it? So for example, if it moved to say 27 degrees C, you might add a little bit of cold water, or you might turn down the thermostat on your water bath. So really important that you look at the number of marks available and explain as fully as possible. Second part of the question says, if it's too cold, how would this affect the results? And we're thinking about kinetic energy. So at 20 degrees C compared to 25, your water molecules are going to have less kinetic energy. They're going to be moving slower. And that means the rate of osmosis is also going to be slower. And therefore, any change in mass that you might get, be that positive or negative, would be smaller in a given period of time. And then a safety precaution question for part C. And it is important that you note that any required practical, you could get asked about safety precautions. For this one, it's talking about cutting. So sensible safety precautions might be carrying the knife pointing away from your body, cutting down onto a non-slip surface, or cutting away from your body. And just to point out again, any required practical, you could be asked about these safety precautions. For the next part of this question, you are given the mean percentage change in mass values for the carrot cubes in different concentrations of sodium chloride solution. And you are also given the standard deviation values. And standard deviation indicates the spread of the data around the mean. So the bigger the number, the bigger the spread of the data. And you are asked to evaluate the student's conclusion that a concentration of 0, so pure water, gives you a significantly higher increase in mass compared to 0.25. And there's some important words here. So they've talked about a significantly higher increase. That word significantly suggests statistical significance. So is there a statistical significance here? And we've been asked to basically evaluate the conclusion. So to what extent does the data support the conclusion? Usually for this kind of question, you need to be including points for and against. So in support of the conclusion and against the conclusion. And the really crucial thing here is the standard deviation values. And what we're looking for is, do they overlap? So if you took water, so 5.8, and you minused 1 standard deviation from that, you would get a value of 4.9. If you then added on plus 1 standard deviation to your 0.25 value, so that would give you 3.9, so 3.1 plus 0.8 is 3.9. So 3.9 and 4.9 don't overlap with each other. You could be asked to work that out using the data in a table, or you could be given the same data in the form of a graph, which is a little bit easier to interpret. So here I've plotted my mean values and I've just plotted standard deviation error bars, so +1 and -1 standard deviation. And then you can just draw lines and you can see whether or not those bars overlap with each other. And here they do not overlap, and that suggests that there is a significant difference between those means. But it only suggests a significant difference. So to be sure, you need to be doing a statistical test. And in this case, we're comparing 2 means. So we've just isolated the data from 2 different concentrations. So we're comparing 2 means. So you would do a t-test if you wanted to prove statistical significance. Part E asks you to explain why the cubes lost mass when placed into 1 mole per decimeter cubed sodium chloride solution. And this is just your classic osmosis explanation. So you need to be talking about the carrot cells having a higher water potential than the sodium chloride solution. Therefore, the water moves out of the carrot cells by osmosis. And don't forget to include that phrase, by osmosis. And then the final part of this question asks you to describe how the student could use the data to estimate the water potential of the carrot cells. And this is a really classic Required Practical 3 question, and it's asking you to use a calibration curve to estimate water potential. And all we mean by a calibration curve in this case is a graph of sodium chloride solution concentration against mean percentage change in mass. And you can then use that to interpolate from 0% change in mass. All we mean by that is read across from 0 until we hit the line of best fit, and then you read off the point that line of best fit hits to read off your sodium chloride solution concentration. And that is the sodium chloride concentration that relates to 0% change in mass. But that is just a sodium chloride solution concentration. It is not water potential. So really important final step is to find a table or graph online to be able to convert that sodium chloride concentration back into a water potential value. Our second question relates to Required Practical 4, which is the membrane permeability practical. And in this case, we have beetroot chunks soaked in vinegar or ethanoic acid, and over time a pink colour has emerged. And the first part of the question asks you to explain why that pink colour has emerged, particularly given that the beetroot has already been rinsed. And that's a really important part of Required Practical 4, that you know that whether you're creating chunks or cubes or cylinders, when you cut the surface of the beetroot, you're going to cut through the cell surface membrane, cut through the vacuole membranes, and if you remember, the pigment is contained within the vacuole. So when you cut through those membranes, pigment will naturally leak out, and you don't want that to be included in your experiment. So you must rinse in distilled water before continuing with your investigation. But that rinsing process has already happened, so where has the pink colour come from? And we're thinking here about the effect of the ethanoic acid, so the low pH on the membrane permeability. But even if you didn't know anything about that, you could assume that because we've got increased leakage of pigment, we have increased membrane permeability. And it's fine to talk about either the cell surface membrane or the vacuole membrane, which is also called the tonoplast, but I would suggest including both. And then the second two marking points are for explaining the effect of the pH on the membrane permeability. And the first point is for talking about the effect on the proteins. So within your cell membrane, you have your phospholipid bilayer, and then embedded in that at various points, you have proteins. And those proteins are important for the structural stability of the membrane. So if you disrupt those, so if you're increasing or decreasing the pH to big extremes, and in this case, we have a lower pH, it's very acidic, we could potentially change the shape of those proteins, denature those proteins, and therefore affect the structural stability of the membrane. The third marking point is for explaining in a little bit more detail why the acidic conditions actually affect the structure of the proteins. And that's because the acidic conditions affect either the ionic or the hydrogen bonds. So you can talk about either, but again, I would suggest talking about both. And those are the bonds between the R groups of the amino acids within the protein that hold the protein together in its tertiary structure. And if you start disrupting those bonds, then you disrupt the shape and the tertiary structure of your proteins, and then ultimately you then disrupt your membrane permeability. The second part of this question relates to the effect of temperature on membrane permeability. You're not actually asked to explain it, but what would happen is as you increase temperature, initially the phospholipids within the bilayer would start to get more kinetic energy, move around more, and that would increase membrane permeability. And then as you increase the temperature more and more and more, eventually you get denaturation of your proteins and you get a more extreme leakage of pigment and increase in membrane permeability. But you're not asked about that. What you're asked about is how you could accurately determine the concentration of pigments within a solution. And in this case, the student has taken cubes of beetroot, they've submerged them into ethanoic acid as before, but in this case, they've put them into a range of different water baths at different temperatures to determine the effect of temperature on that membrane permeability. And they're looking at the colour of the solutions after a period of time in order to determine the pigment concentration within the solution. And they're trying to think about how they could more accurately determine the concentration. They consider using a colour chart, but it's not a particularly accurate method. It's quite a subjective method. So it's a qualitative subjective method. So that means it's non-numerical, so it's qualitative, and it's subjective because it's your opinion really at what point, sort of at which colour matches best to the different solutions. What we want is something more quantitative, so numerical, and more objective, so not purely based on opinion. And the idea that might hopefully spring to mind is using a colorimeter, and you get 1 mark for just using that term colorimeter. And then you need to explain how that colorimeter works. So a colorimeter is a piece of equipment that compares the amount of light that can pass through a solution compared to a standard solution. Now, you often use distilled water as your standard solution because light can easily pass through that. But in this case, we would use ethanoic acid because that is our original solution, so we'd like to compare back to that. So you have a standard solution and you're seeing how much light can get through our new solutions compared to our standard solution. So as we increase the amount of pigment in our solution, what we would expect is that more light will be absorbed by the solution and less light will be able to be passed through. So what you get is either a higher absorbance value or a lower transmission value. And you can use, there's usually a setting on your colorimeter, you can use either or. And those indicate a higher betalain concentration in the solution. And your third marking point is for explaining how do you actually know what the concentration of betalain is in your solutions. So just by looking at the absorbance value or the transmission value, you can have a comparison, so you can see which has a higher concentration and which has a lower concentration, but you can't actually determine the specific concentration of your solution. What we need to do is compare to a solution with a known concentration of betalain. What would be even better is to compare to a dilution series, so to create a range of concentrations of known concentration of betalain and compare the values that you get from those in a colorimeter to the values that you get in your experiment. The third part of this question relates to the effect of ethanol on membrane permeability. And in this case, we're not so interested in the proteins, but on the phospholipids themselves within the bilayer. And ethanol is a non-polar solvent, so it actually dissolves the phospholipids in the bilayer. And you can imagine that's going to have quite a big effect on the membrane permeability. And as we increase ethanol concentration, what happens is we progressively increase the membrane permeability and we get more and more leakage of those betalain pigments out of the cells. And then if we have a little look at question 3, so this one asks us to explain how the structure of a phospholipid bilayer affects the movement of different types of molecule across it. And it specifically tells us not to include reference to membrane proteins in the answer. If we have a little look at the question wording, so it's an explain question, so we need lots of detail. It's asking about the structure and properties of the cell membrane and how they affect the movement of different types of molecule. So when we think about movement of substances, we're thinking simple diffusion, facilitated diffusion, active transport, co-transport. But it specifically says that we should not talk about membrane proteins in the answer. So that rules out facilitated diffusion, active transport, and co-transport, and pretty much just leaves us with simple diffusion. The question also talks about different types of molecules. So it is really important that we are referencing specific molecules within our answer. If we just start by having a little look at the structure of the phospholipid bilayer, because this question doesn't refer to proteins, I've removed the proteins from this diagram, but they would just be embedded within the phospholipid bilayer. But I have shown you the phospholipids with their polar hydrophilic heads and their non-polar hydrophobic tails. And they arrange themselves in such a way that the hydrophobic tails are all facing away from the watery environments outside, and the polar hydrophilic heads are facing towards those watery environments. So you might have tissue fluid on the outside, for example, and cytoplasm on the inside. I've also shown you cholesterol, and cholesterol is important in terms of the permeability and the fluidity of the membrane. Now, just worth pointing out, so in diagrams, often it looks as if the polar heads and the hydrophilic tails are sort of equal sizes, but the hydrophilic tails are the really predominant bit of the membrane that controls what can diffuse through the membrane. So when we're thinking about what can diffuse through, you really need to be thinking that the main part that things have got to get through is that hydrophobic tails in the middle. And if we have a look at the mark scheme, so the phospholipid bilayer allows simple diffusion of non-polar substances, for example, monoglycerides. It's really important that you include the word simple, so it's to differentiate it from facilitated diffusion. And a monoglycerides is a good example, which is what you get when you hydrolyze triglycerides. The phospholipid bilayer prevents diffusion of large polar substances, for example, glucose. I have included the word large there because some small polar molecules like water can actually manage to sneak through. So it is worth saying large polar molecules. And then charged substances definitely can't get through by simple diffusion. So a good example here would be sodium ions. So things like glucose and sodium ions have to rely on channel proteins or carrier proteins to be able to get through. They cannot pass through by simple diffusion. And then a couple of other properties of the membrane that relate to movement. So purely the surface area. So if you've got a bigger surface area of your membrane, you're going to get a higher rate of diffusion. And then finally, the cholesterol that we mentioned on the diagram. So cholesterol affects and maintains fluidity and permeability of your membrane, and therefore will affect the rate of diffusion. The final question provides you with two graphs, and the first of these graphs shows you the rate of uptake of two different substances as their extracellular concentration increases. And the question asks you to state which method of diffusion each substance is moving by and to explain the evidence from the graph. And if we start with substance A, which is our blue line, you can see a proportional increase. And what we mean by that is that as the extracellular concentration is increasing, the rate of uptake is also increasing at a constant rate. And if we compare that to substance B, initially at lower concentrations, we do get a proportional increase at a slightly higher rate. But crucially, at higher concentrations, the rate levels off at a maximum rate. And the reason it levels off is because something is limiting the rate of uptake. And when we're thinking about movement of substances, that something is going to be a membrane protein. So when we think about simple diffusion versus facilitated diffusion, simple diffusion does not require a membrane protein, facilitated diffusion does. So if we have a look at our mark scheme, for our blue line, substance A was moving by simple diffusion, so it doesn't require a membrane protein, and therefore the rate of uptake will be proportional to the substance concentration. And crucial evidence from our graph is that that line does not level off at any point. If we compare that to substance B, we can say substance B is moving by facilitated diffusion, and the reason we know that, so the evidence from the graph, is that the rate of uptake is proportional initially, but levels off at higher substance concentration, and that's really important, that levelling off point. And that's important that you distinguish that from substance A. And then in explanation as well, so the reason it levels off is because the carrier proteins or channel proteins involved in that facilitated diffusion become saturated at high concentration. So you can talk about them becoming saturated or all being in use or working at their maximum rate. The second graph relates to a third substance, substance C, and again you have a graph of extracellular concentration of that substance against rate of uptake, but this time you also have the addition of cyanide. Now cyanide is a poison that inhibits the electron transport chain within aerobic respiration and therefore inhibits the release of energy in the form of ATP. And if you ever get a question that relates to an enzyme inhibitor or a poison that inhibits aerobic respiration, and the question is relating to membrane transport, you want to be thinking along the lines of active transport and ATP being involved. So the answer here is active transport, and the evidence from the graph is that cyanide dramatically reduces the maximum rate of uptake of substance C. So if you look at the yellow line, which is with cyanide, it levels off at a much, much lower level than the blue line without cyanide. And in extra explanation, you know that it must be active transport and it must require energy in the form of ATP from aerobic respiration, because if you take away that ATP, when you inhibit aerobic respiration with cyanide, you dramatically reduce that rate of uptake. So we know that the substance uptake must be an active process requiring energy in the form of ATP. If it was not an active process, if it was something like simple diffusion or facilitated diffusion, adding cyanide would not have an impact on that maximum rate of uptake. And that brings us to the end of this video covering the most common styles of exam questions within the transport across membranes topic. In our next series of videos, we will move on to look at cell recognition and the immune system.